topic is the pistil, megasporangium, ovule, and then the embryo sac. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil, monocarpellary, or may have then more than one pistil, multicarpellary. Means the gynoecium is the female reproductive part of the flower. Androecium is the male reproductive part of the flower. Okay. The gynoecium may consist of the single pistil, means one pistil. It is called as the monocarpellary, or the more than one pistils. It is called as the multicarpellary. Okay. When there are more than one pistil may be fused together and together, or may be free. Okay. When the pistils are fused together, many pistils fused together, it is called as a syncorpus. Okay. When many pistils are free, they are not fused, it is called as the apocarpus. They can give in the second PU definition, or they can give in the NEET examination. Each pistil has a three parts: the stigma, style, and ovary. In the next picture, in the next page, I will show the syncarpus, apocarpus, and in the style, stigma, ovary pictures. The stigma serves as the landing platform for pollen grains. Okay. The female reproductive organ, the gynoecium part, stigma, serves as a landing platform for pollen grains. The style is the elongated, slender part beneath the stigma. Below the stigma, style is present. The bulged part of the pistil is the ovary. The part which is present below the style, it is a bulged part of the pistil is called as the ovary. Inside the ovary is a ovarian cavity is present, that is called as the locule. The placenta is attached inside the ovarian cavity. The placenta is attached inside the ovarian cavity. You can see that ovary present outside, and inside that ovarian cavity is present, and inside that placenta is present. Okay, you can remember like this step by step. Recall the definition and type of placentation that you have studied in morphology of flowering plants lesson. These are the parts of the flower, and then the syncarpus, apocarpus that I explained before. These are the pictures that we can understand by this picture. Very easily we can understand by this picture. This is the a dissected flower of the hibiscus showing pistil. Other floral parts have been removed. Okay. Second one, B is the multicarpellary syncarpus, syncarpus pistillus papaver. C is the multicarpellary apocarpus gynoecium of the Michaelia. A diagrammatic view of typical Hanatropus ovule. Now I will explain the diagrams. This is the stigma, this is the style, and this is the ovary, and this is the thalamus. Okay. And this is the apocarpus. Syncarpus, sorry. B part is the syncarpus. You have to remember the picture. Okay. They will give in the NEET examination. Directly they can give. This is the easy point that you can remember. This is a syncarpus picture. You have to remember that. And example that it is a multicarpellary syncarpus, and this is example of the pistil of papaver. Okay. And this is as a multicarpellary, and this is the apocarpus. So apocarpus, and then the apocarpus of the gynoecium of the Michaelia. These are the carpels. You can see this is a syncarpus. It is like a fused form. Okay. Bulged and fused. It is like a free forms. You can see in this one. It's a carpels. This is the Michaelia. This is the multicarpellary apocarpus Michaelia. Okay. You can remember this as the SP. SP means syncopas per power, and you can remember this as the AM. AM means alarm. We can time. We can say no 9 a.m. 10 a.m. Like this, you can remember this. A means apocarpus ear and gynecium. A means apocarpus and then the M means mycelia. Okay. This is the diagrammatic view of anatropous oval D. D. Now I will explain D. D. This is the xylem part. This is the xylem part. This is the funicle part, and this is the micropyle where the integuments will not fused, and this is the outer integument. This is the inner integument. This is the nucellus. This is the embryo sac. This is the chalagal pole. Okay. This is a diagrammatic view of the anatropous oval. Next one is the arising from the placenta or the megasporangia, commonly called as the oval. Arranging from the placenta means we learned no placenta is present inside the ovarian locules. Arising from the placenta or the megasporangia, commonly called as the ovules. The number of ovules in an ovary may be one, that are the wheat, paddy, mango. Wheat, paddy, mango. Too many, papaya, watermelon, and orchids. Okay. The number of ovules is ovule in the ovary may be one. These are the example for this. These are three examples they are given in NCERT, and these are the example for the many ovules present in the ovary: papaya, watermelon, and orchids. Okay, you have to remember this. And then the megasporangium ovule. Let us familiarize ourselves with the structure of typical angiosperm ovule. The ovule is a small structure attached to the placenta by means of a stalk called funicle. The body of the ovule fuses with the funicle in the region called ileum. Okay, means I will explain this paragraph by the anatropous oval diagram. Okay, the ovule is a small structure attached to the placenta by the means of the stalk called as the funicle. By the means of the stalk called as the funicle. This is the funicle. Mm. This is the funicle. Okay. Okay. This is the meaning of the sentence. And the the ovule is a small structure attached to the placenta by the means of stalk called as the funicle. The body of the ovule fuses with the funicle in the region called ileum. Thus, the ileum represents the junction between the ovule and funicle. Each ovule has a one or two protective envelopes called as the integuments. Integuments encircles the nucellus except at the tip, where a small opening called micropyle is organized. Opposite the micropylar end is the chalaja, representing the basal part of the ovule. This is the explanation that we can easily learn from this diagram. It's a diagrammatic view of that. This is the diagrammatic view of the sentences. Okay. This is the ileum. Ileum is the junction point for the funicle and then the ovule, and this is the micropylar end. Opposite that is the chalagal end, and this is the integument, and this is the nucellus, and this is the embryo sac. You can learn that stuff of the sentences and the paragraph from this diagram. It is very easy to learn. Enclosed within the integument is the mass of cell called as the nucellus. Okay, the enclosed within the integument are mass of cells of nucellus. Cells of the nucellus have abundant reserve food material. Nucellus has a abundant reserve food material. Located in the nucellus is the embryo sac or the female gametophyte. Okay, in the nucellus, embryo sac is present or the female gametophyte is present. It is also, we can see from that diagram, that middle one is the embryo sac. It is also called as the female gametophyte. An ovule generally have as a single embryo sac formed from the megaspore. Okay, an ovule generally has a single megaspore. It is formed from single embryo sac formed from megaspore. Megasporogenesis. The process of formation of megaspore from the megaspore mother cell is called megasporogenesis. This is the important sentence. This is important definition that they can ask in the second P examination. What is megasporogenesis? You have to write that the process of formation of megaspores from the megaspore mother cells (MMC) is called as the megasporogenesis. Ovules are generally differentiate a single megaspore mother cell in the micropylar region. Okay, ovules generally differentiate a single megaspore mother cell in the micropylar region, not the chalagal region. This is the micropylar region. You have to remember that. These are the diagrammatic views. I will explain the diagrammatic views. Micropylar end of the nucellus. It is a large cell containing the dense cytoplasm and a prominent nucleus. It is a large cell means megaspore mother cell is a large cell containing the dense cytoplasm and a prominent nucleus is present. The megaspore mother cell undergo meiotic division means reductional division. What is the importance of megaspore mother cell undergoing meiosis? Meiosis results in the production of the megaspore. Okay, this is the diagrammatic view that we can learn the that you can learn from these diagrammatic views. This is the micropylar end. This is the nucellus. Megaspore mother cell. This is the megaspore mother cell. These are the nucellus. This is the micropylar end. They given in the line of the NCRT that they are differentiate in the micropylar end. You have to remember that. They can ask in the examination micropylar end. This is the nucellus. You have to remember that. And then the megaspore mother cell and differentiation happened here. Micropylar end, nucellus, and then these are the megaspore dyad. Megaspore mother cell divides into the two cells. Okay, megaspore dyads. Okay, they given in diagrammatic diagram also micropylar end. You can remember. And this is the microspore tetrad. These nucellus and you can observe. And this is the division that takes place. Initially, two. After the formation of the eight cells, antipodals and two polar nucleus are formed, and this is the central cell, and this is the egg, and these are the synergid podals, and then the two polar nuclei, and then the central cell, and this is the egg operator. From these eight cells, three cells will form the antipodals. Will come to the antipodal region. It forms the antipodals and two polar nucleus will form, and then the one egg, this is the egg operators, and this is the two synergids, and then this is the egg. Okay, eight cells will distribute like this, and this is the diagram. You they will ask in the that second period examination. You have to write this diagram. Okay, this is the antipodals, and then the polar nuclei, and then the central synergids, and these are the filiform operators that will guide the pollen grains into the ovule. Okay, I will explain about the filiform operators in the next paragraphs. It given in the NCRT. They are given in that 2, 4, 8 nucleate stage of embryo sac or mature embryo sac. This is the mature embryo sac. Okay. Female gametophyte. In the majority of flowering plants, one of the megaspore is the functional, while the other three degenerate. Only the functional megaspore develops into a phyte, that is the embryo sac. Okay. In the majority of the flowering plants, one of the microspore is only functional, while other three will degenerate. Only the functional megaspore develop into the embryo sac. You can understand by this diagram. They gave the diagrams. No, they given the diagram. Out of the four is not mm, mm. Out of the four megaspores, only the one is the functional, three will degenerate, and only the functional megaspore will form the female gametophyte. This method of embryo sac formation from a single megaspore is called as the monosporic development. They can give what is monosporic development. The method of embryo sac formation from the single megaspore is called as the monosporic development. What will be the ploidy of the cells of the nucellus? Microspore mother cell. Megaspore mother cell, the functional megaspore and the female gametophyte. You have to find the answer for this question. I will tell you the answer in at the last video. I will tell you the answer for all the questions that they ask in the middle of the NCRT lines. Okay, let us study the formation of the embryo sac. Let us study the formation of the embryo sac in detail. The nucleus of the functional megaspore divide mitotically to form the two nuclei which moves to the opposite poles, forming the two nucleate embryo sac. Means before I explain with the picture now that only the nucleus of the functional megaspore divide mitotically to form the two nuclei which moves to the opposite directions. Okay, forming the two nucleate embryo sac. Two more sequential mitotic nuclear division results in the formation of the four nucleate and later eight nucleate stage of the embryo sac. I showed now before that two nucleate are there and then the four and it become four and then the eight. It is of the interest to note that these mitotic divisions are strictly pre-nuclear. That is, nuclear divisions are not followed immediately by cell wall formation. Means when the nucleus are dividing by the mitotic divisions, the the cell wall is not formed immediately. After the 8 nucleated stage, cell walls are laid down, leading to the organization of the typical female gametophyte or the embryo sac. After forming the 8 nucleated stage, cell walls will form. Up to that, when there the fourth cell stage, when there is the two nucleated stages, they will not form the cell wall. Observe the distribution of the cells inside the embryo sac. Six of the eight nuclei are surrounded by the cell wall or organized into the cell. Okay. The remaining two nuclei, called as the polar nuclei, are situated below the egg apparatus in the large central cell. This is the very interesting and this is the very important lines. You can learn that after the eight cell stages form, cell walls are formed. Observe that the observe the distribution of each cell inside the embryo sac. Can I show this in the diagrammatic view? Okay. The remaining two nuclei, called as the polar nuclei, are situated below the egg apparatus. You can see, you can understand by this diagram. This one. By this diagram, you can understand. When the nucleus, these are the nucleuses, these are the nucleus. When they are in the 8 cell stage, they lay down and they form the 6 cells, form the cell wall around them. That are the antipodals 3 cells and synergid 3 cells. There are 6 cells. Okay, 2 synergid cells and then the one egg cell, and then the 3 antipodal cells. Okay, these are the nucleus. These are the nucleus. This is the polar nuclei that are laid below the egg apparatus. This is below the egg apparatus. You have to imagine that in the reversal way. They given in the sentence that the polar nuclei are present below the egg apparatus. This is the below the egg apparatus. This is the polar nuclei are not laid by the any cell wall. And then the three, okay, the synergids. Okay, I explain these sentences. There is a characteristic distribution of the cells within the embryo sac. Three cells are grouped together at the polar end and constitute the egg apparatus. At the polar end, three cells are grouped together that are called as the egg apparatus. The egg apparatus in turn consists of the two synergids and then the one egg cell. The egg apparatus consists of the two synergids and then the one egg cell. Okay. The synergids have a special cellular thickenings at the micropylar tip called as the filiform apparatus, which play an important role in the guiding the pollen tube into the synergids. That I before I said about this word, that filiform apparatus. Okay, it is present in the synergids and they have the special cellular thickenings at the micropylar tips. Okay, which helps in the guiding the pollen tube into the synergids. This is the very important sentence. They can ask what is the function of the filiform apparatus in the synergist. In the synergids, you have to write which play an important role in the guiding the pollen tube into the synergids. Three cells are at the salagal end and are called as the antipodal. The cells which are present at the cellulose end are called as the antipodals. The large central cell, as mentioned earlier, has a two polar nuclei are not light by the any cell wall. Thus, a typical angiosperm embryo sac at maturity to 8 nucleated is a and 7 celled. This is a many this this is the mm, this is the very important words. This is asked in the many examination. In the NEET also it was asked.